Hi, in this lecture I will start off with an introduction to the erythrocyte membrane and the learning outcomes for this laboratory. Then I will walk you through the theory behind the three parts of the lab: thin layer chromatography, hemolysis and blood typing. Lastly, we will cover the lab and safety rules, which are especially important for this lab. The erythrocyte membrane is like any other cell membrane and is made up of a lipid bilayer with hydrophobic tails and hydrophilic heads. The membrane consists of various lipids, each modifying the properties of the membrane. Red blood cells, or erythrocytes, have a unique lipid composition. Red blood cell membranes are 1 to 2 times richer in cholesterol compared with any other cell in the body, and approximately 40% of their weight is composed of lipids. The lipid phosphatidylserine in the erythrocyte membrane also plays a role in blood clotting, and the carbohydrates attached to glycoproteins and glycolipids on the surface of the erythrocytes is what determines people's blood groups. Just outside the plasma membrane of this and nearly all cells is a coat of fuzzy material called the glycocalyx, consisting of a myriad of carbohydrate-rich molecules, polysaccharides, proteoglycans, glycoproteins, and glycolipids. Here's just a list of all the lipids that make up the erythrocyte membrane. These lipids all interact with each other to make the cell membrane impermeable. Also note that cholesterol is the most abundant lipid in the erythrocyte membrane, making up almost 25% of the entire lipid content. And here's a small overview of the different lipids from the membrane. Most of the lipids in the erythrocyte membrane have a polar hydrophilic head and a non-polar hydrophobic tail. The hydrophobic tails are buried into the cell membrane, and polar hydrophilic heads interact with other molecules or lipids, for example with cholesterol's hydroxy group, and the rest of the cholesterol structure is buried within the membrane. Cholesterol is the least polar component of the membrane, having only a single polar hydroxy group. Sphingomyelins differ from other phospholipids by the sphingosine group. The goal in this laboratory is to learn about different lipid components that make up the erythrocyte membrane, to learn about different methods and agents that can lyse the red blood cell, and to learn how to determine the blood type of our donor blood, and how blood groups work in general. In the first part of the lab, you will learn how to separate membrane components with thin layer chromatography, or TLC. For TLC, you need a stationary phase made from silica gel attached to a glass plate. Silica gel is a polar substance. The solvent facilitating the lipid separation is the mobile phase, made from a combination of chloroform, methanol and water. The mobile phase is nonpolar. Your samples will be drawn up the plate by capillary action, and their separation depends on 2 points: how soluble your lipid samples are in the mobile phase, and how good the lipids are absorbed by the stationary phase. This is what a TLC plate looks like. Before you add your samples to the plate, draw a line with a pencil on the silica gel approximately 1 to 2 cm from the bottom. Mark the position where your samples will go and leave 1 cm space between them. Once you add your plate to the solvent, the liquid level should be low enough not to directly touch your sample at first. Then you add the lipids, dissolved in non-polar substances, to the position you marked on the plate with a syringe. You don't need much, and these syringes only transfer a few microliters. Wash the syringe in between adding different samples in order to avoid cross-contamination. Once you add your plate to a glass tank with a small layer of solvent at the bottom, you can see that the solvent will migrate up the plate. Non-polar substances will be taken up readily by the solvent and migrate fastest. Polar substances stick harder to the surface of the silica gel and migrate less fast. Once the solvent front reaches about 1 cm from the top of the plate, take the plate out of the tank and let it dry on the fume hood for 10 to 15 minutes. The glass tank contains a filter paper to saturate the air of the chamber with solution. In the second part of the lab, we will have a look at how membrane components can be isolated by destroying the cell membrane. Hemolysis takes place when the erythrocyte membrane is damaged and hemoglobin is released into the surrounding liquid. You will use different methods to carry out hemolysis. First, osmotic hemolysis, detergent hemolysis, and then enzymatic cleavage. Osmotic hemolysis ruptures cells in low salt concentrations. The physiological salt concentration is 145 millimolar sodium chloride, and cells can be lysed by keeping them in a hypotonic environment. When the salt concentration around cells decreases, salt inside the cell is higher than outside the cell, and the cell attempts to compensate by filling up with water. If the salt concentration outside the cell is too low, the cell absorbs so much water that the built-up pressure is enough to burst the cell. This is called hypotonic bursting. Testing for osmotic fragility, how sensitive red blood cells are to osmotic stress, is used to aid the diagnosis of diseases of the erythrocytes, such as spherocytosis and thalassemia, in which the cells have abnormal sensitivity to low salt concentration. Cell membranes can also be disrupted using detergents. Deoxycholic acid is one of the secondary bile acids, which are metabolic byproducts of intestinal bacteria. Sodium deoxycholate is used in research laboratories as a biological detergent to lyse cells and solubilize cellular and membrane components. Another detergent is digitonin, a plant steroid. It has several membrane-related applications in biochemistry, including solubilizing membrane proteins, precipitating cholesterol, and permeabilizing cell membranes. Enzymatic hemolysis is performed by phospholipase hydrolysis. Phospholipase C cleaves off the hydrophilic head from the fatty tail just before the phosphate group. In the final part of the lab, we will look at blood groups and how they can be determined. Overall, there are over 36 different blood group systems, but the two most important ones are the ABO and rhesus systems. The ABO system denotes the presence or absence of A and B antigens on the surface of erythrocytes. There are 4 blood groups in the system: A for carriers of the A antigen, B for carriers of the B antigen, AB if both antigens are present, and O or 0 if neither A nor B are present. In addition, the ABO blood groups are followed by the presence of the Rh-D factor. If the D antigen is present, a person is Rh positive or just positive. If the antigen is absent, the person is called negative. Therefore, if the two systems are combined, a person may be, for instance, A positive, O negative, etc. But what are antigens? They are carbohydrates on the erythrocyte membrane that determine the blood type. Blood groups are inherited from both parents, and the ABO blood type is controlled by a single gene, the ABO gene, with 3 types of alleles. Synthesis of blood group antigens requires at least 2 steps. The first is the synthesis of the H antigen, the structure corresponding to the O blood type. The second is the synthesis of either the A or B structure. The H antigen is generated by addition of a fructose sugar to a terminal galactose. People with type A blood express the A allele of the ABO gene, which produces acetylgalactosamine transferase, or A transferase. The A transferase adds an N-acetylgalactosamine to the H antigen, creating an A antigen. People with type B blood express the B allele of the HBO gene, which produces galactosyl transferase, or B transferase. The B transferase adds a galactose to the H antigen, creating the B antigen. People with type O blood have a mutation that prevents the addition of other sugars to the H antigen. Therefore, they can have neither A nor B antigens, whereas people with type AB express both enzymes and therefore have both antigen A and antigen B. ABO blood group antigens are surface markers on the outside of the red blood cell membrane. Blood group antigens are observed on both glycoproteins and glycolipids. The ABO system is the main blood group system, and the ABO blood group of a person is defined by the antigens on the surface of the red blood cells. Importantly, people produce antibodies against other blood types. So a person with type A blood has the antigen A on their cells, but has the anti-B antibody in their blood plasma. That means that people with type A blood cannot receive blood cells that have the B antigen on it, because their anti-B antibodies would react to it. The reverse is true for type B. People with AB blood have both antigens, but have no antibodies. That means they can receive cells from everyone, since there are no antibodies. People with type O blood have no antigens. However, they have antibodies against both A and B. Therefore, type O blood cells can be given to everyone, but they can only receive blood of type O. For blood donations, only the red blood cells are typically used. So to see if people are compatible, it is important to make sure that the donor does not have an antigen that would react to antibodies in the recipient's plasma. The second major blood group system is Rh-D, or the rhesus factor. Rhesus negative recipients can only receive blood from compatible rhesus negative types. For example, an O-positive patient can receive blood from both O-positive and O-negative donors, but an O-negative patient may only receive blood from O-negative donors. But O-negative people can donate to everyone. They are universal donors because they have no A, B, or D antigens. What happens if something goes wrong and you give B blood cells with the B antigen to a person with type A blood, having the anti-B antibody in their plasma? The anti-B antibodies from the recipient will bind to the B antigen on the donor cells, leading to clumping of blood, called agglutination, which can be fatal. In this lab will we determine the blood type of our donors. In order to test the blood sample for its type, you need to prepare tubes with different antibodies and then add the blood sample to each tube. What has happened when the blood agglutinates? The blood will agglutinate if the antigens on the patient's blood cells match the antibodies in the test tube. A antibodies attach to A antigens. They match like a lock and key, and thus form a clump of red blood cells. In the same way, B antibodies attach to B antigens, and Rh antibodies to Rh antigens. In the test tubes where agglutination has occurred, the patient's red blood cells have been linked together, like a bunch of grapes, instead of floating around one by one. We do not observe agglutination in test tube A, indicating that the patient's red blood cells do not have A antigens, but we do see agglutination in the tube containing B antibodies, which indicates that the patient's red blood cells have got B antigens, thus belong to group B. No agglutination in the Rh tube indicates that the patient's red blood cells don't have Rh antigens, thus the blood is rhesus negative. Therefore the person is type B negative. In order to check if you understood the system, I will give you a minute to figure out which blood cells these three patients have. If you didn't get it right, make sure to revisit the lecture slides or listen to this part of the lecture again. Please take some time to also watch the other videos associated with this lab. The one for performing 1D thin layer chromatography that you can find on Canvas, and also watch the other YouTube videos on blood typing, how to perform ABO blood typing, and the one about osmotic fragility and hemolysis. To check if you've understood everything, you can also take the Erythrocyte lab quiz on Canvas. Please also take some time to familiarize yourself with the lab safety rules. These are very important for this lab and any other. The most important one is to always wear lab coats and gloves to protect yourself. Make sure you know the safety requirements of all reagents that you use and which ones you need to handle with particular care. In this lab, for the TLC part, you will use chloroform as a solvent. Chloroform is very toxic, and you have to make sure that you always work under a fume hood when handling chloroform, and that the fume hood is always properly closed. When working with blood, you should always consider the blood an infectious substance.